Hey guys, so this week, translation to post-translational modifications. Let's get into it. Last week, we talked about transcription and making messenger RNA from our DNA, right? And then we did some post-transcriptional modifications. So splicing is the big one. In eukaryotes, we had a 5' cap and a 3' tail. And after we do that, we're going to send it to a ribosome. And so we need to talk about that. How we're going to actually make our polypeptide chain. And that process is called translation because we're going from nucleic acid, in the case of DNA to RNA, to another nucleic acid. So we're just transcribing. We're staying within the same language, if you will. And then when we're going from a nucleic acid to an amino acid, those are very different things. Now we can use one to tell us about the other. So we're translating our codons from our messenger RNA into our actual amino acid sequence. So let's keep going. Talk about translation without transcription together and without talking about ribosomes. So this should look familiar. We've talked about this before, I think, in this class. If not, we're going to talk about it again. But the idea of having ribosomes and RNA polymerases occurring at the exact same time, that can happen in bacteria. It doesn't happen in eukaryotes because we have that nuclear membrane, okay? But you can have multiple ribosomes on a messenger RNA. So in red, on the right here, is the messenger RNA. And so if you were in a eukaryote, that could head out into either the cytoplasm to a free-floating ribosome or to a free-floating ribosome, and then it would join up with the endoplasmic reticulum, which we're going to talk about that near the end of this presentation. And you can have multiple ribosomes on one messenger RNA. That's absolutely fine. Now, if you are a bacteria and you don't have that nuclear envelope to separate your RNA polymerases from your ribosomes, then you can have them all occur really, really close. So in blue, you can have DNA, and then you can have your RNA polymerase making a messenger RNA right off of that, and you can have your ribosome right next to it. So they're all really, really close. But that only happens in bacteria, or archaea. But when you have multiple ribosomes, that is called either polysomes or polyribosomes. Yeah, so you can have multiple ones. So let's talk more about ribosomes. Ribosome structure is pretty consistent. It's one of those enzymes, if you want to call it an enzyme. It's a little more than an enzyme, but it's basically an enzyme that is just consistent across all living things. So everybody's got a ribosome. If you are a living thing, you have a ribosome, which viruses don't have ribosomes, so maybe they're not living things. But ribosomes, otherwise known as ribozymes, another term for it, are the giant protein RNA complexes that are going to take your messenger RNA and they're going to translate it using their little translator here, your transfer RNA, into an amino acid chain, okay? There are two subunits. There's the small subunit that actually holds on to the messenger RNA, and then there's the large subunit that catalyzes the transfer RNA binding to the codons of your messenger RNA in the small subunit, and it sort of holds onto the amino acid chain and just gives it a little more structure. You can see it's acting much like an enzyme. It's basically a giant complex of enzymes that has multiple functions. Hence, sometimes some people will call a ribosome an enzyme, some people will not. Oftentimes, we will lump ribosomes in as organelles. That's debatable. I mean, you could argue that it's an organelle, but it's way smaller than most organelles. But suffice to say, it's just a giant thing that's going to catalyze reaction. So in that context, I guess it's an enzyme, or a ribozyme, if you will. And so that's the two big things that you need to know. Is the small subunit is going to have the messenger RNA here, and then the large subunit is what is going to facilitate the actual growth of the chain. So let's look at it a little bit more. So here we have our ribosome. So in the green, here we have our ribosome. We've got our large subunit, and we've got our small subunit down here. And you can see we sandwich our messenger RNA in there. And the actual structure of the ribosome facilitates that. It's got like a little groove that your messenger RNA can sit in, and it's got spaces within it where your transfer RNA right here, which has an amino acid attached. We're going to talk a little bit more about that and how they actually get made. But first, we're just going to talk in general what's going to happen. And so we're going to have our tRNA that has an amino acid attached, because they don't have to, but we kind of want them to. They're going to come to the A site. So this is the amino acyl site. Okay, and an amino acyl, that's when we have an amino acid actually attached to our tRNA. We're going to talk a little bit about that in a minute, so don't worry, but that's where that name comes from. It comes from the actual amino acid being stuck on here. Amino acyl is when you take an amino acid and remove the hydroxyl from one end and attach it to the tRNA. So that's what that actually means. Anyway, so it comes into the A site first, and that bonds with, via hydrogen bonding, we have our anticodon on our tRNA, binds to the codon on our messenger RNA. It binds to it and it slides over, so this whole complex slides over. And then we end up in the peptidal portion, or the P site. So there's the A site, the P site, and then the E site. So A site binds, P site is where we actually have the bond formation between amino acids. And so we've got a methionine here, we've got a tryptophan here, and so we're going to actually form these peptide bonds. That's what that's called, a peptide bond. We're going to talk about that in a second as well. And then it's going to slide over one more, and so this is continuous, right? So now something new is in the A site, something new is in the P site, and then this slides over to the E site, which is just the exit site. And that's where you actually lose the tRNA, okay? And so it can go back and become charged is the wrong word, but like it revived with an amino acid attached, and it can go back and be used for something else. And then you're going to just keep on chugging along. You have a start codon, okay, AUG, and then you're going to keep going until you hit a stop codon. Okay? So you have your A, your P, your E site. So some people will call it APE, the APE sites. Other people call it the EPA sites, like Environmental Protection Agency. Whatever works for you, exit, peptidal, amino acyl sites. Okay. Now, when you're actually building it, so we've got our 5' end here again. Okay, so it's same directionality, 5' to 3'. Okay, it was built in that direction as well, and we're going to actually follow along it in that same direction. Now, if we're going to follow along, we're going to build our, here's our start codon, AUG, and that's going to be our N-terminus. Okay, so I'm going to show you what amino acids look like in a second. And so we're going to build our N-terminus, and we're always going to build towards the C-terminus. Okay? Classic question to ask. To which end of the protein that's being made, or the peptide chain that's being constructed, to which terminus are new amino acids added to the growing chain? They're added onto the C terminus. And we need to talk about where these terms come from, N terminus and C terminus. Now, these terms come from the actual structure of amino acids. So look over here with me. Here we have just a classic structure of an amino acid. So amino acids have 2 carbons, 2 oxygens, and a nitrogen, and a couple hydrogens. And they have an R group. So the R group is variable. So normally in chemistry, we just use R as like a replacement variable for any group. Okay? And so in the case of amino acids, there are 20 of them. I will show you a chart in a second. It will be familiar from last time. And you can see, as far as structure goes, R just sticks off here, and then we have our carboxylic acid portion, okay? Hence amino acid. That's the acid portion, and then the aminos part comes from over here. So we got our nitrogen with our 2 hydrogens. We got our nice amine group, and so amino acid. Okay, that's where their name actually comes from. And you'll notice this is an N for nitrogen, and this is a C for carbon. And so there's our N terminus and our C terminus. That's where the nomenclature comes from. And then you have your R group sticking off. And so as we're building, we start with our N terminus. Okay, there's our N terminus. And then we're going to end with our C terminus, sticking off the end here. And in between, we're going to form peptide bonds. Now, peptide bonds, it's a dehydration reaction. If you've taken organic chemistry, this should look super familiar. It's really not that complicated. We're just going to pull this water off, and we're going to form a new bond between this carbon here. So this carbon and this nitrogen of another amino acid. Okay, so we're going to be left with this nitrogen is going to have 1 hydrogen, and this carbon is going to lose its hydroxyl group, and we're left with a nice peptide bond. So that is a peptide bond, and that's what happens in this site, in the P site of your ribosome. There are 20 amino acids, as I mentioned. If you take biochemistry, I know a lot of people take this class concurrently with biochemistry, but some people don't. So I kind of go under the assumption that you have not taken biochemistry and have not talked about peptides and proteins and amino acids yet. So this is a nice little primer for that. There are 20 amino acids. These are the charts that I like to use. I like the circular charts. I don't like the table-based charts. I talked about this when we talked about transcription. Easy question to ask would be to give someone a sequence, a coding sequence, and ask what amino acid chain gets made from that coding sequence. Very, very straightforward. You start in the center and you move out. So in this case, let's figure out. So we've got AUG. AUC, UCG. Let's go with the GAU, even though there's a stop code on here. So GAU, so we would go to, and this is an RNA, so it's because it's got a U, so we go over here. GAU would be aspartic acid, would be the amino acid that gets made. Now, if you take biochemistry, you learn all 20 amino acids that are typical. There are more. There are artificial amino acids. There are amino acids that only exist in certain creatures. There are alternative ways to generate a couple amino acids in humans based on the RNA sequence, but we're not dealing with that. Basically, there's 20 amino acids. That's all you really need to worry about. Now, when you start stacking amino acids together, the order of the actual amino acids, okay, so we've got a nice little order of amino acids here. This would be the smallest peptide in the world. That is called the primary protein structure, or the primary peptide structure. That's just the order of amino acids attached by peptide bonds. And so you can see here, here's just a nice chain of amino acids. There's no folding. It's just one big long chain coming off of a ribosome, okay? Now, when you start folding these things together, and this is what we talk about in biochemistry more, when you start folding those things together, we call that your secondary structure. Okay? And that's generally made up of just alpha helixes and beta pleated sheets. That alpha helices are little helixes, right? So they can kind of stack next to each other, and then beta sheets tend to stack like sheets of paper or sheets on a bed or something. Like they just kind of stack together, so you'd have a few sheets stacked together. That's secondary structure. When I start folding those together, that's when you get your tertiary structure. And so you can see it gets much more complicated. You have a lot more bonding in between your amino acids. And then if you take two separate amino acid peptide chains and stick those together, we call that quaternary structure. Now, there are intermediates, and if you've taken biochemistry, at least with me, you learn that there's tons of space in between here for all kinds of domains and motifs and all kinds of stuff here. But for the most part, this is what typically gets asked. If you were going to take like the MCAT or something like that, these are the four main structures of proteins. On the next slide, there's a few of these. There's like one artist who does these and puts them on Wikipedia. They're really, really good. They're really high quality, and their descriptions are really good. So I put that in the actual slide set because it has a lot of really nice things in it. It goes into a little more detail than we're typically used to, which I like a lot. So for instance, it's coming out of the nuclear pore, which is very interesting. So we have to get our messenger RNAs out of the nucleus. And then there's a 3' untranslated region, and you would have your poly A tail off this end. And there's a 5' untranslated region with a 5' cap. And we're going to talk a little bit more about how those are important in a little while. And then the text here actually goes through exactly what's happening. Now, it's basically the exact same thing that I talked about here, where you have your tRNA bind to the A site, and then you have your P site where the peptide bond forms, and then you have your exit. But if you want to have just another perspective on what's happening, that's why I put this in here. Now, you can see here we've got our ribosomes with our two sides. We've got our large subunit and our small subunit. And we have our transfer RNA. I want to talk a little bit more about transfer RNA. Now, we've learned previously that transfer RNA, among other RNAs, has a powerful set of secondary structures. A lot of hairpin turns. An important idea is that RNA, be it messenger RNA, transfer RNA, any kind of RNA, small interfering RNA, microRNAs, they don't have to be one just long strand that's just stuck there. They fold over on themselves. Okay? And so that's where we get these interesting folding patterns that serve a lot of great functions. That's why, if you look at the actual structure of a ribosome, we're going to talk a little bit more detail about the exact contents that's inside of a ribosome, but suffice to say, it's got just as much, if not more, RNA in it than actual proteins. We're going to talk about that in a second, because RNA can have a ton of structure. So you can imagine as these fold over on themselves, that's kind of dense, and you can fold it into all kinds of really cool shapes based on the actual order of nucleic acids. And that can give you really strong structures that lets you bind to different things. Now, the actual structure of transfer RNA is really interesting. We've got our 5' end and our 3' end. And off of our 3' end, you can see it extends a little bit. That's where we actually attach our amino acid. And the process of doing that is what I wanted to talk about actually next. A lot of genetics courses don't talk about that I wanted to talk about. Not just the tRNA structure, but how we actually make tRNAs. So what we need to do is make an amino acyl group and attach it to our transfer RNA. So how do you actually make transfer RNAs that have the amino acid? Well, you use an amino acyl tRNA synthase. Okay, sorry, synthetase. I'm sorry, it's like the longest word ever. Amino acyl tRNA synthetase. Okay, which is what ARS stands for. Amino acyl tRNA synthetase. I know, it's really long. But basically, how in the world are you going to get your tRNA, so there's your tRNA, this is goopy looking green thing, attached to your actual amino acid? Well, you use ATP. Awesome. And so you've got your amino acid, and this is your enzyme that's going to do it, your amino acyl tRNA synthetase, and you're going to attach it initially to ATP. So we're going to use the little bit of energy that's in our ATP. We're going to break some of those phosphates off. We're going to have an AMP, okay, adenosine monophosphate. We have that diphosphate fall off, and it's attached to our amino acid. Okay, so now it's ready. It's charged, if you will. It's by breaking that bond between these peptides, or I'm sorry, between these phosphates, that allows us to do some work. Okay, and so our enzyme is going to take advantage of that. And so when we're actually going to now, our amino acid has been activated, and we're going to bring our tRNA in. It's going to bind, and we're going to then attach it to this little end right here. Okay. And so we're sort of using the energy from breaking that ATP down to AMP to drive this reaction forward. And then we release adenosine monophosphate, and then you can rephosphorylate it if you would like. And then we release that, and then that's happening all the time. Because think about how many proteins you're actually going to make. We're going to talk about that in a second. You make a ton of proteins. You've got to make, just to make what? Let's say you make 1 million proteins a minute or something. So each of those is going to be made of hundreds, if not thousands, of amino acids that all have their own tRNA. So this process is incredible and massive. I wanted to show one little video just to kind of visualize exactly what's happening when we have translation occur. So I'm going to cut to another North Dakota State University virtual cell. I believe I've shown this before in this class. They'd make really, really good videos, so let's check that out.
Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the amino acyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Those virtual cell videos, they are so good. I like them because they're simple enough, but they do have really, really good quality, and the complexity is high enough as well. So I don't know, I like those a lot. Now, they mentioned something called the release factor, and that's something we haven't talked about yet. So when you actually need to be completely done with your amino acid chain, then you have the binding of not just one protein, but a series of potential proteins that can bind to stop codons. Okay, so let's take a quick look at our codons here. So here are codons, right? We start in the center and move out. And you'll notice there are 3 sites for stop codons. So TAG forms a stop codon, TAA, or in the case of, it would be different for messenger RNA. So UAA and UAG and UGA are all stop codons. And there are proteins that, sorry, what language are we using? There are proteins that will bind onto those and basically just tell the ribosome, hey, stop, release the peptide chain, and it needs to go off. Now, what's really interesting is you can change which proteins are actually associated with that. And by changing that, you can decide different things. And so, like, you can decide if you want your peptide chain to be destroyed. Because maybe you're like, oh, hey, you know what? I don't need this protein anymore, even though I'm already in the process of translating it. So as soon as it gets made, I want to break it apart. So maybe it needs to be ubiquitinated, but you can control that process using the proteins that actually bind to the stop codon. You can also change a stop codon into a different type of amino acid. So we have an alternative amino acid called selenocysteine. It looks just like cysteine, which is an amino acid, but it has a selenium instead of sulfur, and which is just kind of goofy, and it serves just a variety of purposes, mostly for selenocysteine enzymes that just need selenium in them for whatever reason, probably in their active site. And so that happens by changing picking a different protein that binds to the stop codon that is associated with forming selenocysteine. And so instead of stopping the actual amino acid chain, which is what it would normally do, these specific proteins would tell it, hey, add a selenocysteine tRNA-based amino acid, like this dude with a selenocysteine, and have that actually attached to the amino acid chain, and then it keeps going. Really, really interesting. So that's what the release factor is. Two more big things that I wanted to talk about. Ribosome structure is something important when it comes to exactly like the differences between organisms, because everybody has a ribosome, right? Everybody has to make proteins. Bacteria, archaea, eukarya, you name it, it's got to make a protein. There are differences, though. Now, this slide serves 2 purposes. Number one, I wanted to show that you can have some variation between prokaryotes, which is like bacteria and archaea, and eukaryotes, okay? But also, the other purpose is to show you the complexity of what's actually going into the structure of a ribosome. And so we've got lots of protein, so that's what's in yellow here, the protein portions of ribosomes, which is kind of a chicken and egg scenario too, if you think about it, because if ribosomes have proteins, those have to be made from ribosomes, which have to be made from ribosomal proteins. So where did they first come from? Well, that's a good question. That's for a philosophy course. Now, if you've got your proteins here, so we have different pieces of proteins. We've got prokaryotes on the top, eukaryotes on the bottom, and we have our large subunit and our small subunit, right? So we've talked about that, right? We're going to sandwich it in between the two. And we have a ton of RNA. So our ribosomal RNA, so we talked last time about transcription, and your ribosomal RNA gets transcribed inside the nucleolus by RNA polymerase 1 and RNA polymerase 3. RNA polymerase 2 generally makes messenger RNA. And so RNA pol 1 and RNA pol 3 are going to be making your ribosomal RNA, among other things. And you can see they're massive, giant, long RNAs. They look just like messenger RNA. They're made of nucleic acids, just like those are, but they fold over on themselves in very complex patterns. And they are very massive. And what we're going to do is we're going to take those pieces together and combine them with the proteins to actually form our subunits. And so we end up in prokaryotes, we generally end up with a 50S large subunit and a 30S small subunit coming together to form a 70. And then in eukaryotes, we tend to have a 60S subunit and a 40S subunit coming together to form an 80. Now, the S stands for sedimentation, which if you want to, it's a substitute for mass. So when you are looking at proteins, basically what you can do is you see how they they form sedimentary layers when you put them in different density gradients. And so in general, the bigger it is, the larger the S is. So like 70S would be bigger than 30S, of course. But you'll notice 50 plus 30 is not 70, right? 5, 6, 7, so that's 80. It should be 80S, but it turns out the sedimentation units are just kind of goofy like that. They're, even though it doesn't add up correctly, because it's not mass. If it was mass, then yeah, it would add up differently. But if it's sedimentation, well, this actually just sediments. Is that right? It sediments in a different spot, but it's not, like the actual addition. It's fun. You just kind of have to accept it and move on and hope that you never have to do sedimentation. Now, in eukaryotes, we do have a bigger ribosome, and they're showing it disproportionately bigger, if you ask me. But we do have bigger units. Okay? Now, something really, really important that you will encounter in the future, likely, is if you ever hear about like deep sequencing or your microbiome sequencing, which is coming in vogue because it's a lot cheaper to do deep sequencing now. If you need to get your microbiome sequenced, what you do is they'll take it depends on what kind of microbiome you're going to look at. Salivary, they can look in your lungs, they can look in specific tissues, they can look in like your intestines, they can look in sexual compartments, so vaginal compartment usually being the most typical one. And then you will look at the 16S ribosomal RNA subunit. And so that's this guy right here, which we're going to fold together, chop up, and mix it with some proteins to make the small subunit. That is the actual portion that gets sequenced to do microbiome analysis. Because it turns out all your prokaryotes are going to have a 16S subunit, but they're all different enough that you can tell them apart, which is really, really cool. So you can use the same set of primers to amplify the 16S sequence because it's consistent enough across all prokaryotes, but it's different enough to give you some differences. And so that's how you actually figure out, oh, I have a bunch of Firmicutes in my intestines, as opposed to lactobacilli or something like that. So it's very, very cool, but that's where that actually comes from. So that's kind of fun. Now, other prokaryotic differences, so these are your bacteria and your archaea versus your eukaryotes. Eukaryotes, when we talked about transcription, you add a 5' cap and a 3' tail, and those things actually fold over and allow, with the addition of other proteins, they help the ribosome know where to grab onto it. And we call that scanning, which I'm going to talk about that in a second. And the ribosome will actually kind of scan along the messenger RNA and look for, I do have it right there. We'll look for the start codon, okay? But that having the cap and the tail helps it do that. It actually folds it over on itself. Now, prokaryotes are a little bit different. They will not necessarily do the scanning because they don't have a cap and a tail. They just don't need to do that. The caps and tails, not only do they serve the function of like helping the ribosome know where to grab, but they're also really important for getting your messenger RNA out of the nucleus and into the cytoplasm, which bacteria don't have a nuclear membrane. So why would they put these caps and stuff on there? They don't need to. But they're ribosomes, so there's the 16S ribosomal subunit, okay? And it has an anti-Shine-Delgarno sequence that binds onto a messenger RNA that has a Shine-Delgarno sequence. And so it's just a spot that tells the ribosome, because this is basically the ribosome here, right? It's the 16S RNA subunit, which I just got done talking about. And it binds onto that sequence, and it tells it, hey, up a few nucleotides is the actual start codon. And so that's how that works. So it's a little bit simpler of a process as opposed to having a eukaryotic messenger RNA with its caps and tails and all the proteins and enzymes that are responsible for building those things and transporting them outside the nucleus. It just gets way more complicated. So that's kind of nice. It's a little simpler there. And so just to briefly talk about eukaryotic translation initiation, it's a huge complex, okay? So I was saying there's a 5' cap and a 3' tail. I realize this is a really small diagram. What's specifically in here isn't that important, but what I wanted to show is that you've got your 3' poly A tail, right? So there's your tail. You kind of see it there. There's all those little A's, adenines right there. And then you've got your 5' cap, which would actually be on this side, and then it folds over. And so you see the actual piece of messenger RNA that's going to be translated starts with this AUG. I know it's hard to see. It starts with this little AUG here, and then it goes along here until it hits the stop codon. But the actual physical structure of the messenger RNA allows that to occur, which is very, very cool. And there's also So many proteins associated with all of this. What else we need to talk about is the difference between cytosolic ribosomes and the rough endoplasmic reticulum ribosomes. Now, we will often learn early on in cell biology classes and just your classic, just regular biology classes, you learn your different organelles, and you learn that there's the endoplasmic reticulum, which is just a series of phospholipid bilayers, and some of them are rough and some of them are smooth. Now, the rough ER is rough, in quotes, because it has ribosomes attached. And so we need to talk about that. Now, what we're really encountering is the fact that some ribosomes are going to grab onto proteins that need to either be secreted or embedded in another plasma membrane, or they need to I'm trying to think about it. Either secreted or embedded are probably your two biggest ones. So they need to go through the endoplasmic reticulum and into the Golgi, maybe, and then out to the actual cytoplasm, to the cell, not the cell wall, the cell membrane. And have it either stuck there or have that secreted. And so to actually do that, your protein will have a specific sequence. So you can see here, we've got our messenger RNA in green, and that's going to grab onto your ribosome, and it makes your peptide. And that peptide will have a specific sequence, okay, that binds to the SRP, which is a separate protein, okay, for signal recognition particle. It's a particle because it's not just a protein. Just like a ribosome, it also has RNA in there. So that's why it's a little goofy, but whatever. So signal recognition particle, which you don't actually see that often. You don't see things called particles that much inside cells, but whatever. And that's going to bind to the signal recognition particle receptor. Okay, so that's just the kind of bridging the gap between your actual SRP and then the phospholipid bilayer here for the endoplasmic reticulum. Okay, so this is inside the endoplasmic reticulum, this is outside in the cytoplasm. And so your ribosome is going to grab onto your messenger RNA. It doesn't really care. But as the peptide gets made, that peptide might actually pull it towards the actual endoplasmic reticulum. And then that is where you become a rough ER ribosome. It's just, you're being pulled by the SRP binding to the SRP receptor. There are a bunch of other proteins. This is a complex yet still simplified version of what's happening, but basically you're going to then put in the blue here is the peptide sequence, right? And so it's going to actually get translocated into the endoplasmic reticulum to then therefore be folded, like with a chaperone. You're going to have things added to it if you need to be post-translationally modified, which we're actually about to talk about. Now, I wanted to show a video of this process because it's kind of cool. Now, this is one of those videos, the quality is a little bit lower because they're old. They're like 10, 12, 15 years old from textbooks. I think this one's from McGraw-Hill, but somebody like basically did a screen record and posted them because they're really hard to find, these videos. And so we're going to cut to that, and it's going to show us how the endoplasmic reticulum actually like sucks up these proteins.
All protein synthesis in eukaryotes begins in the cytosol, where ribosomal subunits come together and begin translating an mRNA near its 5' end. Soon after the protein starts to emerge from the ribosome, it may become directed to the endoplasmic reticulum, or ER. It is targeted to the ER if it is ultimately destined for incorporation into the ER, the Golgi apparatus, lysosomes, or plasma membrane, or if it is destined for secretion. In mammalian cells, most proteins enter the ER co-translationally. These proteins have an amino acid sequence called a signal sequence, which is typically located at the amino terminus of the polypeptide chain. Signal sequences span about 20 amino acids and include a stretch of hydrophobic amino acids preceded by a basic amino acid. As a signal sequence emerges from the ribosome, it is recognized and bound by a signal recognition particle, or SRP, consisting of 6 polypeptides and a small cytoplasmic RNA. The SRP binds the ribosome as well as the signal sequence, and in so doing, stalls translation. The SRP targets the entire complex to the rough endoplasmic reticulum by binding to an SRP receptor on the membrane of the ER. GTP molecules bind to the SRP and the SRP receptor. The GTP binding triggers the transfer of the signal sequence from the SRP to a membrane channel called a translocon. Hydrolysis of GTP to GDP leads to the dissociation of SRP from both the receptor and the ribosome mRNA complex. Transfer of the ribosome mRNA complex to the translocon allows the signal sequence to interact with short hydrophobic side chains located in the narrow neck of the translocon channel. This interaction opens the translocon by moving the plug away from the translocon channel. As translocation proceeds, the signal sequence is cleaved by signal peptidase, and the polypeptide is released into the lumen of the ER. Thus, the process of protein synthesis directly drives the transfer of growing polypeptide chains through the translocon and into the ER.
That video a lot because it kind of brings a lot of things together, like the structure of a polypeptide chain that's being made, how the tRNA is actually coming into the ribosomes. Super, super cool. Really good video. Now, jumping off of that, so I mentioned post-translational modifications. Now, the very last thing I wanted to talk about is how we actually get variety into our proteins. So your human genome is it depends on who you ask, but 20 to 25,000 genes approximately, which is a lot. But that's not nearly as many proteins as we need. We need millions of different types of proteins. So how do we actually get there? Well, we can have alternative splicing, which we talked about, which is a post-transcriptional modification, right? So I can take my genome, which is DNA, I can transcribe it and then do some post-transcriptional modification to end up with, let's say, 100,000-ish transcripts. And then I'm going to translate that, which would translate directly to proteins, right? So about 100,000 transcripts of messenger RNA can give me 100,000 different proteins. But I can do so many different crazy modifications and foldings of my proteins that I can tenfold increase the actual number of protein species that my cells can have. And of course, complexity goes up from there. And so I wanted to talk briefly, as the last thing to talk about, this is the last slide, about some of those post-translational modifications. If you've taken biochemistry, some of these will feel familiar. If you have not, that's okay. They're really pretty straightforward. Their names explain what's going on. Start in the top left corner, and we're going to go counterclockwise. Hydroxylation, so we're adding a hydroxyl group, so it's like a little alcohol group. So if we added that, that's going to definitely increase your water solubility. I looked up all of these. I just picked some of the big things. Like all of these can do all kinds of different functions and serve different functions for the proteins that they're attached to. So I just picked a couple out. So water solubility is an interesting one. So if I add a hydroxyl group to something, I'm going to be able to increase its solubility in water, which is the medium for all living things, right? A lot of hormone precursors require that. If I actually wanted to get my protein to fold, of course, I could add a hydroxyl group. Now, if I want to get rid of a protein, one great way to actually allow it to increase its excretion or the ability to excrete it like out of the kidneys would be to make it water soluble, right? Because that's what your kidneys are going to actually filter out. So if I have kind of a lipid soluble protein, if I add hydroxyl groups to it, I can increase its water solubility, thus increasing its ability to be secreted in water. Now, what's kind of interesting is the actual hydroxylation process requires iron, so you got to have your iron in your diet, and it requires vitamin C or ascorbic acid. And so if you have low levels of vitamin C, which is called scurvy, you may have heard of that, like pirates and stuff. So if you're not getting your vitamin C, you're not going to be able to hydroxylate your proteins, which is going to have a lot of really big problems. Especially a lot of hormone precursors are hydroxylated, and so that's going to really screw you up. Now, something else you can do, you could add a methyl group. Okay, so a carbon with a couple hydrogens on it. Of course, most of these are going to have like protein-protein interactions. I just threw those on there because, I mean, obviously, if proteins need to interact with each other, sometimes they need a methyl group, sometimes they don't, it doesn't matter. But one of the big ones for this is histones. So epigenetic control and how tight or loose your chromatin is controlled by either methylation or acetylation, to simplify it, of your chromatin and of the histones that actually form your chromatin. The general rule is if your histones are methylated, that tends to make them tighter. Okay, so they can have more interactions with those methyl groups. They kind of tightens up your chromatin, makes it harder to transcribe from that. And if you acetylate, which is down here, so let's just jump to that. So if you acetylate, that means you're adding an acetyl group to your proteins. And what that can do is it can loosen it. And so you can control how tight your chromatin is by methylating it and how loose it is by acetylating it and different combinations thereof. And so that's one of the big job of acetylation, at least in the context of this course. Now, something else, you could add lipids to it. Okay, so you could add lipids to things, and what's that going to do? Well, the big one that comes from is if I take a lipid, like a big fatty acid, and I stick that onto, which fatty acid, just big long carbon chain, basically. If I stick that onto a protein, that's going to allow the protein to be embedded in a membrane more easily. It may be involved with some of the signaling, and every single one of these can get really, really complicated real fast. So if one of these sounds really interesting, you can dig a little bit deeper. But that kind of makes sense because phospholipid bilayers have fatty acids in them. And so if you put a big hydrophobic region on a protein, well, that's going to make it easier for it to hang out in hydrophobic areas. I picked, it has disulfide bonds. So disulfide bonds just occur between two cysteines. So they have sulfur that stick out. I guess methionine has sulfur too, but usually we think about it as two cysteines, and they form just a special bond between the two sulfurs. And that's going to just increase the strength between two protein strands. Okay, so you end up with these disulfide bonds here. But it's not really something you add to it per se. It's just going to add to your structure. So let's keep going. So I always say this wrong. I haven't really actually heard a good way to say sumoylation, whatever. Sumos are small ubiquitin-like modifiers. So ubiquitin, so let's look up here first. So ubiquitination is usually understood as a way to destroy a protein. So if I want to get rid of a protein, I can ubiquitinate it. And ubiquitin is just a really small chunk of protein that you can tack a bunch of them onto another protein and cause it to be degraded. That's what we typically think about it. And so the proteasome complex, which is what chops up proteins that are bad or misfolded or we just don't need them anymore, usually we would ubiquitinate, and that attracts it into the proteasome for a breakdown. Now, it has a ton of other functions too, so I don't want you to think that ubiquitination is solely for destroying proteins. That is absolutely not true. It has lots of other functions. You can read about them, but destruction is the one that we usually think about. Now, sumoylation, small ubiquitin-like modifier proteins. So you tack those on. They are ubiquitin-like proteins, which is a whole class of proteins. They're just sort of like ubiquitin, but they don't really serve to destroy stuff. So from what I could find, they're basically for nuclear import. So if I need to bring a protein into the nucleus, which is atypical, you wouldn't normally want all your proteins rushing into the nucleus, and so you need to control that actual import. And the export of things out of the nucleus is also very controlled, but the import is as well. And it also allows for protein stability, which is important. You don't want them falling apart. Same thing for glycosylation. So I can take sugars, just like lipids, I can tack sugars onto stuff, and that can do all kinds of things. Stability is the big one, but also signaling molecules, because adding sugars to a variety of molecules can really change how they interact with their receptors. And so I could have one protein with a bunch of different lipidations or methylations or glycosylations, and that can change how it interacts with other receptors, which is really, really cool. Last but not least, because I don't know, I just wanted to say that, we have phosphorylation. So typically we would think about this in terms of activating something or taking a protein and you throw phosphate on there with ATP and that kind of charges it up or powers it. You're going to see that with tons of transport proteins. We phosphorylate things all the time. That's like the entire function of the entire class of proteins called kinases. That's what they do. They phosphorylate stuff, usually other proteins. And so actually adding a phosphate onto things can change it. It's going to change the charge. It's going to allow some energy to sort of enter that process. You can use it to do a variety of things. nice and glossed over. But like I said, if you want to dig deeper into any of these post-translational modifications, you can. But the main point is that you can take a single protein and add tons of different modifiers on it and absolutely completely change how it interacts, how it folds, its co-interactors. Super cool stuff.